Ladies and gentlemen, good evening. My name is Attila Magyari Sáska. I am a student at Babes-Bolya University and Matthias Corvinus Collegium. Welcome to today's Transylvania Lecture, which is a platform created for the local public with the aim to discuss topics relevant and interesting, not only for our Transylvanian society, but for the larger intellectual sphere as well. Today, it is my honor to present to you our dear guests, to whom we are grateful for accepting our invitation. On one hand, Dr. Dina Zielinski, an American scientist living in Paris. A molecular biologist by training, she was inspired to learn bioinformatics to bring biological data to life. She completed her bachelor's degree in biology and French at NYU, followed by a master's degree in bioinformatics from Université Paris-Cité. She has completed a joint PhD in genomics and biomedical informatics between Harvard Medical School and Sorbonne University. Between 2019 and 2023, she was a researcher at INSERM, the French National Institute of Public Health, where she developed predictive models to improve diagnosis in transplant patient care. She currently leads computational biology R&D at White Lab Genomics, a deep tech startup that aims to mitigate risks and accelerate the development of genomic medicines. Her work has been profiled by The Atlantic, BBC, Forbes, NPR, The Pathologist, The Scientist, STAT, The Wall Street Journal, and Wired, and her TEDx Vienna talk on DNA storage, published on TED.com, has received nearly 2 million views. On the other hand, her partner in conversation, Ákos Bálint, an IT specialist from Marosvásárhely, Turgumúres, with more than 20 years of experience in this field. He was a founder of Ebola in Romania, collaborated with international companies, and in 2013, he co-founded Upforte, a mobile app development agency. Besides the IT industry, he has been working as a trainer and workshop facilitator. The exploration of DNA as a potential solution for digital data storage offers an intriguing intersection between biology and technology. As nature's most ancient and efficient storage device, DNA holds the promise of transforming the way we think about data preservation in an era of exponential information growth. By leveraging the storage capacity and longevity of DNA, we may unlock new methods for encoding and maintaining digital data in a format that has stood the test of time. This discussion invites us to consider the vast possibilities at the intersection of biology, technology, and data storage, shedding light on how the oldest storage device in nature might inspire innovative solutions to modern challenges. Might there be a world where DNA even replaces the iconic floppy disk symbol as the universal image for data saving? Dina, Ákos, the floor is yours.
Thank you. Can you hear me? Using these? Okay. Can you hear me now? Yes. Okay. Thank you for the invitation. It's great to be here. And looking forward to a lively discussion, which Ákos will lead.
Sure.
Now to kick things off, so as we all know, we've already told you the punchline, what nature's oldest storage device actually is. So you're all aware of where we're going here. But basically, our world today is driven by information. The amount of data that we generate every minute is astounding. And we all now have essentially supercomputers in our pockets with our smartphones. And no, most of us don't actually think of where all of this data, all of our tweets, all of our YouTube videos, this video is going to go on YouTube, that's going to take up data storage space. Most of us don't actually think about where this data is being stored. But it's in the cloud. Most of us don't even think about what that means and the repercussions. But the truth is, we are running out of storage devices. We are hitting a global data storage crisis. So as data generation and consumption grow, we have to update our data storage vocabulary. How many people here are familiar with IT in general and bits? Okay, great. We've got a lot of pros in the audience. So I don't have to go too much over this little review. But the point is here that most of us work with data in the megabyte, the gigabyte range. Some, maybe if you're a data scientist or work in information technology, you might work in the terabyte range, but it probably costs quite a bit. But the truth is we are entering what's known as essentially the zettabyte era. We now have generated digital data, all of humanity, that is in the order of zettabytes. And we're actually running out of storage vocabulary. So the highest approved international unit is known as the yottabyte. So we have to decide what will follow the yottabyte. Some people have comically suggested the hellabyte, but then who knows where we'll go with the language there after we get past the hellabyte. So data generation has increased exponentially just in the past 10 years, with more data generated in just the past few years. And we're, as I mentioned, we're officially in this zettabyte era. And I actually, every time I give a talk about data storage, I have to update all the figures that I use in the talks. Because just last year, we were still talking about being in the 60 zettabyte range, but it's estimated that just by next year, we'll surpass 175 zettabytes of digital data globally around the world. So data generation, as I mentioned, is outpacing our ability to actually store that data. We're literally running out of devices. And data centers, where all this data is stored and processed and transferred, require immense amounts of energy. And as our data storage consumption grows, so too will that data storage and energy requirement. And right now, data centers account for a small percentage of global energy consumption, but that's only going to increase. So it's in our nature to record human history. We used to do it on stone tablets. Now we use our smartphones. Everywhere we go, we're documenting everything. And fortunately, methods have improved, so we can record more and more data. But basically, the improvements that we have made since even just the 1990s, something that used to be super impressive. I mean, back in the 90s, when Bill Gates here, the co-founder of Microsoft, posted this image of himself on a stack of papers holding up a CD-ROM. He was really proud of this feat. And now most of us find this pretty comical. We don't find this very impressive because does anyone here use a CD-ROM? Does anyone here have a way to read a CD-ROM? Okay, we have a few in the audience, but most of us have no way even to recover data from a CD-ROM unless you go to like an internet cafe or talk to the two guys in the audience. It's really a continuous struggle throughout most of human history to find ways to document and reliably store data. Turns out stone tablets and paper are the most reliable way to store data. We've spent a lot of efforts in just the past 100 or so years on improving and miniaturizing and reducing the cost of storing digital data. The problem with this is that every time we invent a new storage medium, we have to invent a new way to read it and a new way to write it. And there's this issue of data migration. So every time we update a new device, we update the readers and we have to migrate the data. And that's due to longevity and also to changing technology. And so if you have data stored on one medium, you have to invest the time and the money to migrate that data to the new technology. So DNA is essentially migration-free. DNA is not going to change. The medium is what it is. And as long as we're around, it will never be obsolete. So DNA doesn't have this migration problem. Now, new technologies have increased substantially. As I mentioned, paper is still one of the most reliable forms of data storage. We know this from books. They can last quite a long time. We found ancient hieroglyphics on papyrus from ancient Egypt. Most books will last easily hundreds of years as long as they don't get, they're not involved in any natural disasters or caught in a flood or anything like that. But basically, most of these improvements in data storage capacity come at the cost of longevity. And so you can see there that DNA, it is unmatched by any of these man-made devices in both longevity and in storage density. So this is cheating a little bit because DNA it's a molecule that basically is quite sensitive to humidity and to UV rays. So on its own, DNA is actually quite sensitive to the environment. But fortunately, we have ways where we can store that DNA reliably. So one such way is storing it in what's called silica beads. So these are just glass beads. You lose out on some of that density, but you can reliably protect that DNA easily for literally thousands of years. Whereas in most of these cases, your data is going to last tens of years at best at most of these man-made devices. So DNA is also incredibly durable. So the oldest DNA found in nature is from more than 2 million years ago. So scientists discovered DNA from the permafrost in Greenland from more than 2 million years ago, and they were able to extract DNA from this sample from more than 130 different species of animals, and they were sort of able to recreate this ecosystem from 2 million years ago. No single man-made device could store information even remotely closely to that kind of time frame. And DNA is incredibly dense. I mean, a density that I think most of us have a hard time wrapping our heads around. Storing data in DNA is not new. Nature's been doing it for literally billions of years. To give you an idea, if we take the DNA in our cells, so we have 3 billion base pairs of A's, T's, C's, and G's in all the cells in our bodies. And basically within each cell is the rough equivalent of 1.5 gigabytes of data. And we have more than 30 trillion cells in our body. Now, if you were to consider all of the world's data stored on all of the world's devices, that represents less than 1% of all the data in a single human person. So what that means is we would have no way to store all of that data. Now, it's obviously redundant, right? I'm cheating a little bit. Our genome is the same across all cells. But the scale of that density is just impossible to even imagine. We have absolutely no way to store that level of information. So in theory, every living thing is a DNA storage device. Some are more sophisticated than others. I used to have another politician up here, but I couldn't stand to look at him, so I changed it. But so, most of these examples of living storage devices vary in complexity. So for example, a banana has about 500 million bases, so letters of DNA. A penguin has about 1 billion, whereas we have 3 billion. So actually not quite a huge difference. Just the point here being that you have different scales of density, but it's still quite high. So I have to always show this slide because one of the key questions that I always get is when and how can I store information in my own DNA? From storing, I don't know, family secrets or photos in your own DNA and passing it on. I highly recommend not doing so. There's a lot, are there any biologists in the audience? So there's a lot in our genome that we still don't understand, but that doesn't mean that it's not actually serving a purpose. So if we go ahead and insert DNA sequences, we might disrupt some important functions. DNA is storing a lot of important information. It's what basically provides the instructions to tell a single cell to develop into that 30 trillion celled organism. So best not to mess with it. But the way we do this is we synthesize DNA in a tube. So you can much more safely store data in DNA in a tube. So DNA is truly the ultimate storage medium. It's been optimized, as I mentioned, for literally billions of years. But it really is only recently that it's been explored as a viable digital data storage solution. So pun intended here. Just going to go through a bit of history. So this is one of my favorite photos. This is photo 51. It's the first ever image of the structure of DNA. And this was published in 1953. And just 3 years later, the first ever commercial hard drive was released by IBM. And it could hold a whopping 3.75 megabytes, which is like one MP3 song. And I think it costs about 10,000 US dollars. So, you know, not actually very useful for anything we would want to do today. But the point is that, you know, we've seen storage and computing evolve quite a lot since 1956. And our understanding of DNA and of information technology have sort of co-evolved in many ways. However, as I mentioned, using DNA as a storage medium has really only been explored in the last 10 years or so. So this is the progress that we've made in data storage in the last 65 years or so. So on the left is that hard drive that I showed you, and on the right, with a 50 euro cent piece for scale, you have basically, you know, an example showing how well we've succeeded at miniaturizing data storage. And in that same amount of time, we've learned to read, write, and copy DNA from that moment in 1953 where we just figured out what DNA structure even looked like. And this is the progress that we've made in DNA sequencing just in the past 20 or so years. So on the left, you have this enormous DNA sequencer. And on the right, here with a 50 euro cent piece for scale, you have literally a handheld DNA sequencer. And this sequencer can actually generate sequencing data in real time, as opposed to the original first human genome took many years to generate. And we've gone from days to hours to literally minutes to be able to sequence DNA. So the idea of using molecules to store digital information is not new. It was actually conceived of by Richard Feynman, an American scientist, back in 1959. And then it was Michael Niemann in 1964 that actually proposed this idea of using DNA to store data. And then it was this crazy looking guy known as Joey Davis at MIT that did the first ever proof of concept of storing information in DNA back in 1988. So very briefly, this was a very small scale proof of concept experiment. He took an image known as Microvenus. It's basically an ancient rune. And he essentially he encoded the pixels essentially in that picture into 35 bits, and then converted that bit sequence into a small string of DNA. And then he essentially pasted that into DNA that could be inserted into bacteria. So then that message was essentially encoded in that bacteria. Now this is very small scale. He wasn't able to read that information back. Basically the technology was not quite at scale to read and write DNA, but it was still a really nice proof of concept of the possibility. And from 1988, and there was not a lot of exploration in the DNA storage space. It wasn't really until the 2010s or so that the technology was able to sort of meet the theory. So here if we look at, do we have any scientists, or anyone who uses PubMed for papers? Okay, we have a few people familiar. So PubMed is essentially a website where all scientific publications are stored. And so this is just to show you the number of publications in DNA storage published in the last 30 odd years. And you can see that the vast majority were just in the last 5 or so years. And so, we've made enormous progress in that short amount of time. So storing data in DNA seems really far-fetched, but we've seen how long it took us to go from that almost room-sized hard drive to having a literal supercomputer in our pocket. So it's really just a matter of time before the technology can catch up with the theory. So how much can we actually store in DNA compared to human-made devices? So if we take the global data sphere, which again, that's all the digital data in the world, it's about 120 zettabytes. that would require about 7 kilograms of DNA. And so I tried to find something typically Romanian that I could figure out the weight of. I didn't know the weight of goulash, for example. So I found these beautiful decorative wooden eggs, and I think you can estimate that they're about 50 grams each. And so to store all of the digital data in the world, you would need about 140 of these. I imagine that's a little too much for decorating, but when it comes to data storage, it's actually not too bad. On the other hand, if we take DNA's biggest competitor, magnetic tape, so magnetic tape was first used in the 1950s for computing and storage. And most people don't store data on magnetic tape, but any big data company will. For example, if there's a data loss, I know at Google there was a major Gmail loss and they had to restore user accounts using magnetic tape. But essentially this is DNA's biggest competitor because it is quite affordable, easy to update, and it has continued to increase in storage density over time. But instead of 140 decorative eggs worth of volume essentially, you would need 2 billion 90 terabyte tape cartridges at 100 euros each to store the global data sphere. So as you can see, DNA wildly outperforms any human-made device in terms of density. And the other point is that this tape would need to be updated every 10 or so years. So you have to pay that amount every 10 or so years when you want to update the data. Whereas, I'll get to the cost in a bit, DNA is still wildly expensive, but those costs are continuing to come down. So the storage of digital data beyond timeframes of 50 years is incredibly challenging. The amount of data stored on today's highest density storage magnetic tape, if stacked to the moon, would essentially fit into a shoebox if you stored that same amount of data on DNA. So again, I mean, I think I've made my argument pretty clear that DNA is unmatched in density. However, while DNA is about 5 orders of magnitude more dense than magnetic tape or other media, it's also 5 orders of magnitude more expensive. So even more so than that crazy expensive 100 euros for 2 billion magnetic tapes. So it's also nearly 200 times slower to write data on DNA than on magnetic tape, but it can be stored literally thousands of times longer. So it's going to come down to a cost versus benefit. And obviously, the idea is not to store data that you need quick access to, but mostly to what's known as cold data. And most of the data that we retain, about 60% or so of retained data is actually cold, meaning that we don't have to constantly access it. So we can write it once, store it on DNA, put it away somewhere cold and dry, and it'll be safe for hundreds, maybe even thousands of years. So, okay, so I think I've convinced you of how reliable DNA can be, but how do you actually store stuff in DNA? So there's a few key steps. So one is to start with encoding. So essentially anything that you can store on normal man-made media can be stored on DNA. So basically any digital data. The only difference is that you have to convert those zeros and ones into the DNA alphabet. So if you remember from biology class, A's, T's, C's, and G's make up the DNA alphabet. So you essentially convert those zeros and ones into A's, T's, C's, and G's. And then once we have that file, we can send that off to a company. There's DNA synthesis companies that will literally print out the DNA and put it in a tube and send it right back to you. And then, of course, you can store it if you want to store the data over time. And to recover the data back from DNA, you have to sequence it. And then you have to decode it back from DNA to zeros and ones. So obviously, this is a bit oversimplified, but these are the key steps to a DNA storage system. So as I mentioned, it's a bit more complicated than what I showed. It's not really just about converting zeros and ones to A, T, C, and G. This is a key step, but it is the easiest step. And the fortunate thing is that any sort of encoding schemes that we apply to digital data, we can also apply to DNA. And so the coding scheme is actually quite simple. Because you have 4 letters in the DNA alphabet, you can store up to 2 bits per letter. And so essentially 00 becomes A, 01 C, and so on. And so, not only is DNA incredibly dense and durable, we also know how to write, read, and copy. We can do so many things with DNA, pretty much everything that we can do with a standard storage medium. And so I'm going to just for those of you who aren't familiar, I think most of you are pretty high-tech here, but just to give an example of what it actually means to encode data. So if we take the simple example of Cluj, if we convert those letters into binary, this is essentially the easy step. We can then apply any standard coding operations, which is essentially just adding some redundancy, error correction codes. Essentially, you can take your data. If we want to take this message here, Cluj, and we want to send it over an information channel, or we just want to store it, you have to add essentially some redundancy of extra zeros and ones. And as you add zeros and ones, obviously, that reduces the amount of data you can store. And then the easy step, again, is just converting those zeros and ones into DNA. So that's how you would spell Cluj in DNA. So back in 2017, I co-authored a method that improved the density and error correction of DNA data storage. So what we did is we essentially used a more efficient algorithm that was developed in 2002. So as I mentioned, the same algorithms that can be used with binary data can also be applied to any data stored in DNA. And so as scientists, we can be very serious, but we also like to have fun. And one of the fun parts of this study was choosing which files we were going to store. So one was an old operating system called Calibri. Anyone heard of it? Okay, we've got one. And we have Claude Shannon, the father of information theory, a manuscript that he wrote. A zip bomb, which for those of you who survived the 90s, this was a virus that you could send around that would essentially keep successively unzipping files until it crashed your computer. Really nice. And then one of my favorite choices was one of the first films ever made, which was The Arrival of a Train. And on the bottom left, you have what's known as the Pioneer plaque, which was included in the Pioneer spacecraft, so that extraterrestrials would know what a typical human being would look like and our location in the galaxy. And finally, an Amazon gift card. I think there were $50 on it. And so some guy managed to decode it, and he used the $50 to buy a super nerdy book on Amazon. And so essentially, we can compress those files. It just becomes digital data made up of zeros and ones. And then we convert, as I showed you, using that coding scheme, we convert those zeros and ones to C's, T's, and G's, and A's. And then we can essentially send what's literally a file, just an Excel file, for example, of A's, T's, C's, and G's to a company. And in this case, we sent it to a company called Twist Bioscience out in California. And I think about a week and a half or so later, they shipped this tube back. And in that tube is basically a small film on the bottom. So we dry out the DNA. So the DNA doesn't like moisture. So you want to keep it cold and dry. And all of those files were essentially in that little tube. So to get them back, you literally just add water and sequence it and then decode. And so this is the code that we developed for our study. So this is just to show you. Obviously, I sped up the video a little bit here. But this code is freely available on GitHub if anyone wants to try it. Essentially showing that you can run a simple data storage solution on your MacBook. So this was done in just a few minutes on a standard MacBook Pro. And once we do the data encoding, we get the output file, which is essentially just a string of letters of DNA. And that's the file that we send to a synthesis company. And so in that tube, that was about 2 megabytes of data. And this was, again, back in 2017. and that cost about 7,500 US dollars. So as I mentioned, not very viable for storage solutions. If you really want to make sure your data is highly preserved and it's highly valuable, it can be done if you have the budget. And that cost is going down. We're actually getting down to about $1,000 per megabyte range. So things are moving pretty quickly. But writing DNA is prohibitively expensive. So the reading part, the sequencing, has dropped enormously, exponentially. But we're still waiting for those DNA synthesis costs to come down. But every year there's new advances. And so just to kind of give you an idea, so the invention of the printing press in the 15th century had revolutionized the way that we record and distribute information. And there's sort of a similar revolution happening in biology. So most of you, thanks to COVID, are familiar with PCR. So this allows us to copy DNA molecules over and over again. So as I mentioned, ordering that original tube of DNA is extremely expensive. But once we have that tube, we can essentially just add water to dilute it, and we can make copies over and over and over again. And we demonstrated in that study back in 2017 that data can be copied and recovered even after being copied trillions of times. We can still recover the data off of that DNA without any errors. And PCR is quite cheap. It's on the order of a few euros or US dollars, as opposed to, 7,500 for 2 megabytes of data. And so as I mentioned, there's this sort of revolution happening. We've turned to nature for a data storage solution in DNA. In a similar fashion, there's actually enzymes that exist to copy DNA. And so we can use those enzymes to synthesize DNA in a tube. So the typical method of synthesizing DNA requires a really wasteful, harmful, and costly chemical synthesis process. But in just half the time, and with much less harm to the environment, we can use these enzymes in a tube to actually write out the DNA. And so there's companies and scientists that are actively working on this to bring down the costs, because this is essentially the major bottleneck that's preventing us from consistently using DNA as a storage solution. So bringing down these costs is incredibly important, not just for storage solutions, but also for synthetic biology in general, for developing gene therapies, vaccines, not to mention DNA computing. So DNA is not just a storage medium. It actually can be used for computation. So the first ever DNA computer was developed in 1994, and this was by an American scientist called Leonard Adleman. And he essentially used DNA and an enzyme in a tube. So again, some strands of synthetic DNA. And he was able to solve very quickly what's known as the traveling salesman problem, which is essentially, it's called a minimum weight Hamiltonian cycle. That's the technical term. And the point is to find the shortest route for a salesman through all the cities that he has to visit, or all the houses he has to visit. And so he was able to do this in a tube with DNA in a matter of minutes. And this is something that even the most advanced supercomputers couldn't do in that amount of time. And again, the only bottleneck here to making DNA computing a reality is that this was a very small scale experiment, and Adleman was very aware of that. In order to scale this up, it would be prohibitively expensive because you have to synthesize all of those DNA sequences. But again, as those costs come down, DNA computing will become a viable solution as well. So then that gets us to what should we actually store in DNA? Now, we're constantly recording with our phones, and I think most of us could agree most of the videos and the photos and the emails and the text messages, we don't go back and look at the vast majority of those. But the thing is, we didn't, when our ancestors were writing on papyrus or stone tablets, they didn't have to filter that information. And we could tell a lot about ancient civilization from things like the Rosetta Stone, from cave writings. digital data is incredibly fragile. I mean, all of our recorded human history could easily disappear. And so deciding what should be stored, I think, is a huge challenge. And hopefully the cost will come down so that we don't have to be too picky. But as of now, the best solution for DNA is archival storage. So another option for DNA, again, so we can compute, we can store data. There's another approach called DNA of things. What this means is you can essentially store information in DNA in a physical object. So to give you a use case example, this could be used, for example, for supply chain tracing. So you can essentially store DNA in a glass bead, like I mentioned, and you put that in a 3D printing polymer, and then you can insert that in an object. And then if you're concerned about, the fidelity of that object, for supply chain, for example, you can just break off a piece of that object and sequence it to make sure it's actually what it's supposed to be. And then one of my favorite examples is, how many people here are Massive Attack fans or have at least heard of the band Massive Attack? So yeah, they actually collaborated with one of my colleagues at ETH in Zurich, and they decided for the anniversary of their album Mezzanine, they did just that. So they stored the DNA in silica beads and put it in spray paint and made art with the Mezzanine album stored in DNA in the spray paint. So this was pretty cool. And so another example I like, again, it's really hard to choose what we should actually store. And I think once the costs come down, hopefully those choices won't be as difficult. But one of my collaborations was actually with a artist. I love the convergence of science and art. And we essentially stored the first ever museum in DNA. And so by museum, this was essentially just a representation of a physical layout of a museum. And we stored that in DNA in a tiny tube. So the museum is in that small vacuum tube there. And in that vacuum tube, the DNA can last for hundreds, even thousands of years easily. And my favorite example as a scientist is that website PubMed. Any scientific discovery is built on previous scientific discoveries, which have taken decades. And so if we lost all of that information, we would have to start from scratch in many respects. This scientific literature is critical. So this is one example that I think that we could use to store on DNA, is to back up all of that scientific literature and put it essentially somewhere safe, cold and dry. And so these are all just kind of proofs of concepts and kind of crazy ideas and, bands with too much money and too much time. But there is actually a great example that currently exists. And so this was a world's first. The Declaration of the Rights of Man was stored in DNA and is actually in one of those little vacuum tubes that I showed you. And it's stored in Paris in the French National Archives. Now this is a document that was written after the French Revolution in 1789, establishing human rights. And so it's a document that's critical for humankind. And so this is a great example of something that we could back up so that it's safe from our global storage crisis. And just for a local example, I saw that most of the records in your archives here are being digitized. Most of them are not yet digitized. But once they are digitized, this is another example of historical data that could be stored on DNA to store family records, important history that's important to Romania and Hungary and this entire region. And finally, so as I mentioned, these are all possibilities that we could potentially store, mostly archival data that's important for humankind as we're waiting for costs to come down. And this is essentially a sort of proof of concept. It's a global seed vault. So here's where A lot of seeds are stored in case there's some sort of environmental crisis. They're backed up so that if we need to, we can regrow different plants and fruits and vegetables. And I imagine a similar solution for data. We could store data in DNA and keep it along with these important seeds in the seed vault in Norway. So there are significant hurdles to actually making DNA a viable storage solution. But as I mentioned, it's only a matter of time. And we're looking at maybe within the next decade, you'll start seeing data consistently stored on DNA. And as I mentioned, we don't have that migration problem. As long as we're around, DNA will never be obsolete like floppy disks and CD-ROMs. Thank you.
We thank you. Can you? Is this working? Okay, no. Just keep it close enough, but not too close. Find the right direction. Oh, okay. So that was really interesting. And before you would switch to a little bit of perspective to my side, from let's say for the IT. As we saw, as we learned here, that there are much more technical guys in the audience than biologists. So let's widen a bit the perspective, and can you give us a picture about this genetical engineering landscape? So because I assume that this is storing the data in DNA is not the only research direction in this area, and I guess that there are a lot of others, and there's a close interaction between them. So if you can tell us a few words about it.
Yeah, sure. So just to give an idea, so as I mentioned, the cost of synthesizing DNA is extraordinary. And mostly that's because we've been synthesizing DNA like that for decades. As scientists in a lab, we need to know that the DNA sequence that we order is exactly what we order, because we're essentially manipulating those molecules in the lab, testing different modifications to genes, changing the DNA to see how it changes gene expression. And so it's really critical that those sequences are precise. One advantage of DNA storage is we have information theoretic approaches that allow us to use essentially a quick and dirty synthesis. We don't need the DNA synthesis to be quite as good. We can use error correction and more digital information theoretic approaches to handle a less clean synthesis process. But in terms of synthetic biology, it's limitless, the things that we could potentially do. But obviously we're most familiar with, for example, the COVID vaccines. So these were RNA vaccines. And I'm currently working in the field of cell and gene therapy. And so these are essentially genetic medicines. And these are typically used to treat incredibly rare, incurable diseases. So sorry, I'm a biologist, and once I get going, I'm hard to stop, so please feel free to stop me. But there's about 7,000 rare diseases, and only about 5% of these actually have an approved treatment. And that's because they're incredibly difficult to treat. However, many of you might have started to hear a lot about cell and gene therapies. There's therapies that were recently used to cure advanced melanoma. You have gene therapy that has cured sickle cell disease. We have gene therapies that can cure blindness, restore hearing. And essentially this means synthesizing DNA and putting it in a vector, which is actually typically a virus that can no longer infect, and delivering that to the affected tissue. And so this is one of the key uses for synthetic DNA.
I see. Well, when I first read about genetic surgery and gene surgery and editing, genetic editing, I thought about tiny scissors and I don't know, clips, and so tiny tools that you manipulate the gene. And only later I understand that this is some chemistry. So this is not mechanical manipulation, but chemistry. And what is the history of this well, science, bioscience?
So we're talking about gene editing. So CRISPR,
so,
for example.
Yes.
Yeah, I think probably most people have heard of CRISPR. And this was discovered, I think, in the 1980s, actually. It's essentially an immune defense system that exists naturally in bacteria. And scientists realized that they could actually take that technology, so essentially use this method to edit our own genomes. And so most of you have probably heard about it in more recent years. There's actually approved drugs on the market that use CRISPR technology. And so the advantage of CRISPR, so the approach that I was describing for gene therapy, we essentially deliver the functional copy of a gene, so that synthetic DNA essentially, into the affected or diseased tissues in a person's body. But that DNA doesn't integrate into your genome. And so it just exists typically in a circular form in the nucleus of your cells. And so the advantage of CRISPR is that you can permanently change the DNA. So the advantage there is that with gene therapy, we think it tends to persist for 10, 20 or so years, but there's just not enough cases and not enough data to really know if gene therapy will provide a lifetime single-dose cure. And the problem of redosing gene therapies is that we typically use what's called viral vectors. And so we know a lot about viruses since the pandemic, but the vast majority, like 99% of viruses, don't actually cause illness. And one of those viruses is known as adeno-associated virus, or AAV. And these viruses don't cause illness in humans, but they are exquisite at getting into cells. And so we can essentially take the shell of that virus and we can insert that synthetic DNA, and that virus will go to the proper tissue to essentially deliver a functional copy of a gene in a sick person. And again, the disadvantage is that it's not necessarily permanent, whereas CRISPR allows you can use the same approach, you can deliver the CRISPR machinery to the affected cells, and then it will modify the DNA permanently. The risk there is that we could have what's called off-target effects. And so you might accidentally end up editing some important parts of the genome. And so this is something that we have to be cautious about. But it's drastically improved just in the last 10 years, for example, with base editing. So this is a much more precise version of CRISPR.
Wow, that's incredible. And returning to the data storage, so how mature is the technology now? I mean, the challenges are more scientific ones or more engineering challenges?
So more engineering, and that's one of the reasons why I'm not currently working, I'm not focusing on DNA storage. This was a super fun study that we did back in New York, and I still keep up with the literature, I review papers, we're working on a textbook, but it's essentially become an engineering problem, and I'm a biologist, and my passion has always been to cure diseases. And so that's where I'm focusing my efforts. And there's people who are much more qualified at improving the chemistry synthesis and just the engineering challenges of developing DNA storage solutions.
So it's in the applied research phase, I guess. And are there any big companies interested in this technology?
There's a lot of companies, the government too. For example, I know the US government is highly interested. They put out major grants. Microsoft has a big DNA storage investment. There's also companies like Twist Bioscience, Cache DNA. I mean, all over the world, there's new companies popping up working on improving DNA data storage solutions.
And do you think this speed of reading and writing barrier can be broken at a certain point?
Absolutely, 100%. So we've seen it with DNA sequencing. So the Human Genome Project was completed in 2003. That's when we put together the first ever full human genome. And this cost a few $1 billion, US dollars. And now we can sequence an entire human genome for less than $1,000 just in 20 years. And again, we haven't really focused on reducing synthesis costs because again, it was suiting our needs, and when we order synthetic DNA in a lab, we don't need such high volumes as we do for storage systems. And so it's not actually insanely expensive. Usually if I order DNA in a tube, it's on the order of tens of dollars or euros for our needs in the lab. And so it's only recently that we're focusing on finding solutions to reducing synthesis costs. So I think it'll happen probably in the next decade.
So maybe within 10 years we might see the DNA in our laptop?
I don't know about that. I still think it will be mostly archival storage and cold data. For example, if you take Amazon Glacier, if anyone stores their data in the cloud, this is essentially a way to store data that you don't use very often. You know, maybe you have legacy data in your company, or some old financial records, surveillance data, who knows. You can essentially store it for much, much cheaper than typical data storage, so data that you need to access constantly. It's, again, they call it Amazon Glacier because it's essentially storing cold data. So you pay a premium once you want to access the data. But as long as you don't touch it, it's, you know, pennies for gigabytes or something ridiculous.
Yeah. So I was thinking about what kind of use cases might exist. And as the copying the data, replicating the data is quite cheap, as you said. So maybe it can be useful in cases when you have cold data and you need a lot of copies of that data. Say like the Windows install CD back in the 90s. But in one of your presentations, I watched on YouTube the videos, and you were talking about this visualizing or modeling or representing the black hole. So there was a lot of measurements around the world, and they took the data, put together a lot of hard disks. So they were piled up in a table, and maybe those data can be copied, so that amount of data and used by multiple labs to do the same research again, to bring the study further, maybe. So can you imagine?
So for that example, for those of you who remember, the first ever image of a black hole was published back in 2019. I think it was April 2019. And essentially, this was data collected from telescopes all around the world. And so they essentially, to share all of their data, they had to put it on external hard drives and ship it around the world because I guess FedEx or whatever postal service they were using had a faster data transmission rate than actually sending it over the internet. But to store it on DNA, yes, you could absolutely, once you make that initial library, it would still be insanely expensive to do so. But let's say once those costs come down, you can store those satellite images, those basically on an initial pool of DNA, and then you can make copies very cheaply using PCR. And then, yeah, ship that around the world in a little envelope.
And keep it forever. Yeah. And okay, so I've learned only now that this how these DNAs are synthesized, and I can understand the advantage of synthetic DNA comparing with the living DNA because, well, it doesn't change. Well, I suppose it doesn't changes over the time, while a living organism, there are changes in the structure. So how stable is this synthetic DNA comparing with the living one?
Yeah, this is a great question. So when you have DNA in a tube, it's just DNA alone, just strands of A, T, C, and G. And there's nothing in there. If you have UV rays or moisture, this can cause changes in the DNA and introduce errors. So it can cause breaks in the DNA strands. It can convert different letters to other letters. And that will make the process of reading that data back from DNA very challenging. So in our bodies, we have enzymes that repair DNA. So that's happening all the time. We all have rogue cells in our body that have mutations in them. Unfortunately, sometimes those cells go rogue and develop too many mutations for the cells to handle. But that's where the information theoretic approaches come in. And so we can encode the data in DNA in a way that allows us to reliably recover that data. So using good error correction codes and really focusing on the information theoretic side. But again, as long as DNA is stored cold and dry, it can last hundreds, thousands of years. We've seen it. One of my favorite examples is Ötzi the Iceman. So he was found in the Alps, the Tyrolean Alps between Austria and Italy. And then this led to Austria and Italy fighting over him. But it turns out so they sequenced his DNA, and it turns out he has cousins in Italy and Austria. But the point is, this guy who's, from, I think, 10,000 years ago, we were still able to find living relatives. And usually when I show Ötzi, I'll show a picture of a broken smartphone. We could get more information from Ötzi the Iceman than from a broken smartphone.
Okay, and on the other hand, I guess, with synthetic DNA, you don't have the constraint of a living organism. So let's say, can it have any length, synthetic DNA?
Another excellent question. So a lot of the technologies these days are really limited by the length. So in theory, yes, you could sequence DNA of any length. The problem is once you get past like a few hundred bases, so DNA letters, you start to introduce errors. And this is every step of the way, from writing the data to reading the data. So synthesizing DNA, we can only do up to a few hundred letters. We now have methods called long-read sequencing, so you can sequence literally tens of thousands of letters in a row. And the error rate is higher than these other methods, but it is possible to sequence extremely long fragments. The thing is, though, that when you store DNA over time, it eventually breaks into smaller fragments. And so this small read sequencing is not necessarily the worst thing. But part of the decoding that data back requires piecing those sequences back together. And so if you have things, if your information is in just one sequence, it just makes the decoding much easier.
I see. Okay. Now, after you presented the technology, it seems this storing data in DNA, it seems very straightforward, the idea itself. And well, yes, there's a structure that was designed for data storage, and we have the technology to use it as a data storage. But this DNA computing, that's something mind-blowing for me. So how does it work?
So, I mean, DNA computing is still in its infancy. I think the best example was the one that, the original one, the first proof of concept shown by Leonard Adleman in the 90s. And the reason why we haven't really advanced is because if we were to carry out that experiment on more than, let's say, 7 cities, it would just be prohibitively expensive. But another use case is for, you could use DNA molecules essentially to generate random numbers. Random number generation is really useful in data encryption. So that's another great use case. But again, synthesis costs need to come down.
So it's a bit similar to quantum computing, that it's very good for a certain class of problems, I guess.
Yeah, for highly complex, time-consuming problems that require a lot of parallelization.
And which are difficult to compute linearly, but
exactly. Yeah.
I see. Okay. Now we are talking about data, the stability of the data, the write and read speed. Another very important aspect is the security of the data. And of course there are the standard data protection methods, what we use, encryption and whatever. Are there any medium-specific possibilities with DNA?
Absolutely, yeah. I mean, so one, you can access, you can essentially hack a DNA sequencer to access the data that's being sequenced, just like you could access like a network. Another challenge is that you can essentially synthesize harmful sequences, so pathogens, viruses. The thing is, it would require quite a lot of molecular biology skills to take those synthetic sequences and actually create a virus, but it can be done. So, of course, for any good technology, there's, of course, people out there who could conceive of using it for nefarious reasons, but that is one of the biggest concerns. And a way around that is to sort of introduce a screening protocol in any DNA storage solution, where you would essentially screen the generated sequences for any known pathogens. But again, it would require quite a lot of molecular biology knowledge, but not all scientists behave so.
Not for everyday people, okay. And wow, and is it possible to add searchability to the DNA storage? So to read any sequence?
Yes, so like any good data storage solution, you need what's known as random access, especially since, so when you sequence the DNA, when you're recovering your data, normally if you recover, like you open a file on your computer, you're not destroying the data. This is not the case for DNA. If you take a sample of the DNA, which is essentially just molecules in water, you're literally destroying that in the process of sequencing. So there's the advantage of copying via PCR. You can make kind of an infinite copy, but yeah, you're essentially destroying the information as you're sequencing it.
I see. So I never thought of this aspect of it. Okay. Now from the IT perspective, I mean, this DNA and all this stuff is just it's a black box. I mean, I want to send the data in and I want to take the data out. And basically, what I understand that we have to wait 10 more years and we will actually have this black box on our desks, more or less. Now looking from another perspective, so I assume that a lot of computation is used in this kind of research. I intentionally did not name it artificial intelligence because there's a lot more beyond AI, not just AI, and then now the hype is with the generative AI, but I guess a lot of algorithms are used and a lot of computation is used. And if you can tell us about these, I know you're a biologist, not a data scientist, But
I'm a computational biologist. I use some AI methods in my analysis. In the company I'm working for, actually, we're developing AI models in order to essentially accelerate the development of these genetic medicines that I talked about. And these models are very data hungry, especially when it comes to deep learning models. The challenge with, if we were to store that data on DNA, it would be incredibly expensive, and to train these models, you need to be able to access the data. So there's a high latency in actually accessing the data from the DNA. But potentially, maybe, eventually, once it becomes more affordable, or just storing at least a backup copy of that enormous amount of DNA, because as we continue to develop these models, we're going to also continue to generate an enormous amount of biological data, because we're really only scratching the surface of the amount of data that we can use. And these models are mostly useful for highly complex, vast amounts of heterogeneous data. And so the more we have, the more challenging it will actually be. But in terms of storing on DNA, it's still quite challenging. And as I mentioned, within a decade, yes, we'll probably be storing data on DNA somewhat on the regular, but mostly archival data. But I think most of you won't even know it. So it will essentially be, you'll be storing, maybe Amazon Glacier will invest in DNA data storage, and you won't even know that your data is being stored in DNA. It's unlikely to be something that's in your home or a thumb drive that is connected to your laptop, at least not in the next decade.
Okay, and then we have a lot of students here in the room, and some of them has IT knowledge, and some of them can have or have already biology knowledge. And how can you connect these two skills so to go further in your direction?
So I'm a trained biologist. I studied hardcore biology at university. And then I started working in a lab at MIT, and I was the only biologist. And this was when genomics was really advancing incredibly rapidly. And I essentially got just really tired of waiting for all of the programmers in the lab to analyze my data. I couldn't stand it. And so finally one day I sat down and asked them to show me. And so this just started with learning what the command line was and how to run different commands, and then shell scripting, and then R, and then Python. But it essentially came out of frustration that I learned to become a computational biologist. The other advantage is that I can work anywhere in the world now. I'm not required to go into the lab every day to check on my cells. Cells are incredibly needy, like harder than taking care of a cat. And so it's actually quite nice to be on the other side. I transitioned slowly from being about 50% computational and at the bench, and now all of my work is fully computational. But it's good to have an understanding of both sides. So I've worked with scientists who come from the computer science side of things and learn the biology. I knew the biology and have learned the computational side of things. So I've mostly learned by doing. I did a master's in bioinformatics, but I'm not a computer scientist. But I know enough not to do anything stupid.
So you write Python scripts in your job?
Well, now GitHub Copilot does that for me.
Copilot in general.
Yeah.
Okay. Well, Dina, thank you very much for sharing all this knowledge with us.
Thank you for all the fantastic questions and for the invitation. So this is actually my third time in Cluj. I'm a big fan. So thank you again for yet another warm welcome.
Well, yeah. Then see you next time.